understanding the breakthrough in rectal cancer treatment and the Dostarlimab's promise. Rectal cancer, alongside colon cancer, forms a significant portion of colorectal cancers, affecting millions worldwide. While advancements have been made, the search for more effective and less invasive treatments continues. Enter Dostarlimab, a cutting-edge therapeutic solution targeting a specific subset of rectal cancer patients, those with mismatch repair-deficient dMMR tumors. This drug's performance in clinical trials offers a glimmer of hope for a group previously facing challenging outcomes. The focus of Dostarlimab. Dostarlimab is designed to target and treat mismatch repair-deficient locally advanced rectal cancer. This condition, characterized by a failure in the DNA repair system, makes tumors more susceptible to immunotherapy. Clinical trials have shown promising results, with a significant portion of patients achieving complete response without the need for conventional treatments like chemotherapy, radiation, or surgery. However, it's crucial to note that this effectiveness is specific to dMMR rectal cancers, which represent approximately 10% of all rectal cancer cases, a relatively small but significant subset. Clinical trial, insights and statistics. In a groundbreaking study, Dostarlimab demonstrated a 100% clinical complete response rate in patients with dMMR locally advanced rectal cancer. This achievement marks a potential paradigm shift in treating this cancer subset, suggesting a move towards non-invasive management options. Such results spotlight the importance of precise genetic testing in cancer treatment, ensuring that targeted therapies like Dostarlimab reach those most likely to benefit. Imagery and interpretations from the trial. The document provided in the link below this video contains several images and tables that elucidate the drug's impact and the study's methodology. 1. Evolution of endoscopic and radiographic response. Images showing the dramatic reduction in tumor visibility, highlighting Dostarlimab's effectiveness over time. Figure 1. Evolution of endoscopic and radiographic response. Description. This figure shows endoscopic evaluations, T2-weighted MRI of the rectum, and FDG PET scans for 2 representative patients at baseline 3 months and 6 months. Interpretation. For patient 2, an endoscopic complete response and a near-complete response on rectal MRI at 3 months, and a clinical complete response at 6 months are demonstrated. For patient 9, both endoscopic and radiographic complete responses are visible at 3 months. The arrows in the images identify the tumor at each time point, highlighting the significant reduction in tumor visibility and metabolic activity over time, indicating the treatment's effectiveness. 2. Biopsy specimens before and after treatment. Microscopic views revealed the transition from visible tumor cells to a state with no viable tumor cells post-treatment, emphasizing the drug's potent anti-cancer activity. Biopsy specimens of the rectum before and after PD-1 blockade. Description. This figure presents hematoxylin and eosin-stained biopsy specimens before and after treatment. It shows viable tumor cells surrounded by necrosis before treatment and a cellular residual mucin pools after 3 months of treatment. Additionally, it estimates the percentage of viable tumor cells over time in 10 patients. Interpretation. The progression from clearly visible tumor cells to almost no viable tumor cells post-treatment underscores the dramatic impact of Dostarlimab on tumor cells. The presence of acellular mucin pools after treatment indicates the absence of viable tumor cells, suggesting a strong response to the PD-1 blockade. 3. Immune contexture changes. Illustrations of how Dostarlimab influences the tumor microenvironment, promoting an immune response capable of targeting and eliminating cancer cells. Immune contexture changes after PD-1 blockade. Description. This figure includes multicolor fluorescence images showing the levels of PD-L1 protein, CD8 plus T lymphocytes, and CD20 plus B lymphocytes at various stages of treatment. It quantitatively analyzes the changes in these markers in the tumor and normal epithelial cells and in the stromal tissue compartment. Interpretation. The data demonstrates how PD-1 blockade therapy modulates the tumor microenvironment over time. An increase in PD-L1 protein and CD8 plus T lymphocytes suggests an enhanced immune response against the tumor. The presence and increase of CD20 plus B lymphocytes, especially forming tertiary lymphoid structures, indicate a robust adaptive immune response, potentially contributing to the treatment's effectiveness. These figures collectively illustrate the profound effect of PD-1 blockade therapy on mismatch repair-deficient locally advanced rectal cancer, showcasing the therapy's potential to induce significant tumor regression, modulate the immune response, and possibly obviate the need for more invasive treatments like surgery. These visuals provide concrete evidence of Dostarlimab's action mechanism and its transformative potential for patients with dMMR rectal cancer. According to the document, the treatment with Dostarlimab, a PD-1 blockade therapy, specifically targets mismatch repair-deficient locally advanced rectal cancer. This specificity is crucial because mismatch repair deficiency is a condition in which the cancer cells have mutations that prevent them from correctly repairing the DNA inside them. This type of cancer is more likely to respond to immunotherapy treatments like PD-1 blockade due to its high mutational burden, which makes the cancer cells more recognizable to the immune system. Therefore, the groundbreaking results presented in this document pertain only to a certain group of rectal cancers characterized by this mismatch repair deficiency. It does not apply to all forms of rectal cancer. Mismatch repair deficient cancers are a subset of colorectal cancers that represent a distinct pathogenetic group, which is particularly sensitive to immunotherapy treatments like Dostarlimab. This specificity highlights the importance of genetic and molecular testing in cancer treatment, allowing for more personalized and effective treatment plans based on the individual characteristics of a patient's cancer. A call to action. The promising results of Dostarlimab in treating mismatch repair-deficient locally advanced rectal cancer represent just the tip of the iceberg. As research progresses, the potential for similar breakthroughs in other cancer types and subtypes grows. To continue exploring and sharing these advancements, we invite our viewers to subscribe and like our content. Your support fuels our mission to research and create content that benefits patients, researchers, and the broader community. Together, we can illuminate the path to more effective cancer treatments and a hopeful future for those affected. Conclusion. The journey from understanding the complexities of rectal and colon cancers to the development of targeted treatments like Dostarlimab reflects the power of precision medicine. By focusing on the genetic and molecular underpinnings of cancer, researchers are unlocking new, less invasive treatment paradigms. As we delve deeper into the capabilities of such therapies, the importance of community engagement and support becomes ever more apparent. Join us in this endeavor to shed light on the cutting-edge research that promises to redefine cancer treatment and offer new hope to patients worldwide.